Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
A living cell, when viewed through a conventional light microscope, presents a frustrating paradox. It is a world of immense complexity, a bustling metropolis of organelles and molecular machinery, yet it appears as a mere ghost, a faint, translucent outline adrift in a sea of bright light. Its internal secrets remain securely veiled by its own transparency. The very property that defines it as living, its aqueous, unstained state, renders it all but invisible. For decades, this presented biologists with a stark choice to observe a cell's structure by killing, fixing, and staining it, or watch its ghostly, featureless silhouette move, guessing at the dynamic processes within. This fundamental barrier to knowledge was dismantled by a Dutch physicist whose intellectual toolkit was forged in the seemingly distant fields of optics and statistical mechanics. Frits Zernike, a professor at the University of Groningen, approached the biologist's dilemma with a physicist's eye for the unseen properties of light. He recognized that while a transparent specimen like a living cell has a negligible effect on the amplitude or brightness of the light waves passing through it, it does impose a subtle yet crucial alteration. The light that traverses the denser parts of the cell, such as the nucleus or mitochondria, is slowed down slightly. It emerges delayed, its wave pattern shifted in time relative to the light that passed through the surrounding medium. This change is called a phase shift. To the human eye, and to any standard microscope, this phase shift is completely imperceptible. Our visual system registers only amplitude and frequency, brightness and color. The universe of phase information was, for all practical purposes, a hidden dimension. Zernike's profound insight was to find a way to translate this invisible phase information into a visible amplitude signal. His solution, developed around the 1930 and formally detailed in a 1934 publication, was a phase contrast method. It was an idea of stunning elegance, achieved through the introduction of a single, massively designed optical component to the phase plate. This small, transparent disc is placed at a specific location within the microscope's objective lens at the back focal plane where the diffraction pattern of the specimen is formed. On this plate, Zernike etched a ring-shaped groove or deposited a thin layer of material. This ring corresponds precisely to the path of the undiffracted background light that illuminates the specimen. The function of this phase ring is twofold. First, it selectively alters the phase of this background light, typically retarding it by a quarter of a wavelength, which corresponds to a phase shift of pi over 2 radians. Second, the ring is often coated with a light-absorbing material to dim the background light. Now, when the two sets of waves of the background light altered by the phase plate and the specimen light diffracted and phase shifted by the cell's structures recombined to form the final image, a remarkable transformation occurs. The engineered phase difference between them leads to powerful interference. Regions of the cell that produced a phase lag now appear dark against a brighter background, or vice versa. The invisible phase shifts have been converted directly into visible contrast. Suddenly, the ghost in the machine materializes. The nucleus resolves into a sharp, dark sphere. Mitochondria appear as distinct rods, and the delicate membranes of the endoplasmic reticulum become visible. The intricate, real-time ballet of cell division, once a matter of inference from static, stained slides, could be witnessed directly. The immediate impact of this invention was revolutionary, even if its initial reception was lukewarm. The optical firm Carl Zeiss at first showed little interest, a historical detail that offers a quiet lesson in how truly disruptive ideas are often perceived. For the biological community, however, once the technique became available, it was akin to being granted a new sense. It allowed for the detailed, continuous observation of living processes without the artifacts and cellular death associated with staining. Cell biology, microbiology, and hematology were transformed. Researchers could now study the effects of different substances on living cells, watch bacteria divide, and observe the behavior of parasites within host tissues, all with unprecedented clarity. Zernike's work, while brilliantly original, stood upon the shoulders of giants in optical theory. His entire conceptual framework is a practical and ingenious extension of the diffraction theory of image formation developed by the German physicist Ernst Abbe in the 1870s. Abbe was the one who first articulated that microscopic imaging is a two-stage process. First, the specimen diffracts the illuminating light into a spectrum of waves, and second, the objective lens collects these diffracted waves and makes them interfere to reconstruct the image. Zernike's genius lay in his deep understanding of Abbe's theory and his realization that one could actively intervene in that second stage. He was, in essence, engineering the interference to reveal the information he wanted. Another clear intellectual antecedent was the Foucault knife edge test, a method used by opticians to detect tiny imperfections on the surface of telescope mirrors. The knife edge physically blocks some of the diffracted light, making surface flaws visible. Zernike himself acknowledged that his thinking about phase contrast grew from his analysis of the diffraction phenomena underlying the Foucault test. The ripples of Zernike's innovation spread outward, directly influencing the next generation of optical invention. His method demonstrated that the phase component of light was a rich, untapped source of information. This principle directly inspired Georges Nimorsky to develop differential interference, contrast, or DIC microscopy in the 1950s. DIC microscopy uses prisms to split light, pass it through adjacent points in the specimen, and then recombine it, visualizing the asterisk gradient asterisk of the phase shifts. This produces a pseudo-three-dimensional, bas-relief image that is exceptionally useful for studying the surfaces and outlines of living cells. In the world of biology, pioneers of cell biology like Robert Chambers, a master of micromanipulation, could now see the cellular structures he was probing with his microscopic needles. The technique became a cornerstone, a standard method for anyone working with cell cultures. Today, its relevance endures. Phase contrast microscopy remains a fundamental tool in laboratories worldwide. It is the default method for routine checks of cell cultures, for clinical analysis of blood and other bodily fluids, and for a vast range of research in developmental biology and neurobiology. It provides the essential visual context for more complex fluorescence microscopy techniques, allowing researchers to see the overall morphology of a cell while simultaneously tracking fluorescently tagged proteins within it. Zernike's elegant solution to the problem of transparency did more than create a new tool. It fundamentally altered our relationship with the microscopic world. allowing us to watch life unfold undisturbed. This profound ability to render the invisible visible, to conjure substance from shadow, rests upon a beautiful and precise foundation of wave mechanics, a physical and mathematical architecture. We shall now explore the ability to visualize transparent biological specimens, a seemingly insurmountable challenge for early microscopists, yielded to Frits Zernike's ingenious invention phase contrast microscopy. This achievement, a testament to his profound understanding of wave optics, rests on the precise manipulation of light phase, a property often overlooked in simpler analyses of light intensity alone. Zernike's insight lay in recognizing that the subtle phase shifts induced by transparent objects, while imperceptible to the naked eye, encoded valuable information about their structure and composition. His solution elegantly transformed these imperceptible phase variations into readily observable amplitude variations, rendering previously invisible details strikingly visible. The core principle hinges on the wave nature of light. Light, as a wave, possesses both amplitude and phase. Amplitude corresponds to the intensity of the light wave, determining its brightness. Phase, however, represents the position of a point on the wave cycle, a subtle characteristic that impacts how waves interfere with each other. Transparent objects, unlike opaque ones, do not absorb light. Instead, they alter the phase of the light wave passing through them. These phase shifts, often minuscule, are the key to visualizing transparent structures. A phase shift of a quarter wavelength, for instance, represents a significant alteration within the wave's cycle. Zernike's phase contrast method cleverly exploits the phenomenon of wave interference to amplify these subtle phase variations. The method employs a special optical element, a phase plate, strategically placed in the microscope's optical path. This phase plate, a marvel of precision engineering, introduces a controlled phase shift to certain components of the light wavefront. Specifically, it selectively alters the phase of the diffracted light, the light scattered by the specimen, while leaving the undiffracted light, the light passing through the specimen unaltered, largely unchanged. Consider a simplified scenario. A transparent specimen, such as a living cell, introduces a phase shift of approximately a quarter wavelength to the light passing through it. Without the phase plate, the diffracted and undiffracted waves would superpose, resulting in minimal observable contrast. Zernike's phase plate, however, introduces an additional phase shift to the diffracted light, shifting it by a further quarter wavelength, thereby increasing the total phase difference between the diffracted and undiffracted components to a half wavelength. This carefully engineered half wavelength phase difference is crucial. Waves with a half wavelength phase difference interfere destructively, meaning their amplitudes subtract. The phase plate, in essence, converts the phase difference into an amplitude difference. The resulting interference pattern reveals the specimen's structure with remarkable clarity. Areas of the specimen that introduced a phase shift appear darker or brighter against the background, depending on the precise phase shift introduced by the phase plate. This is the essence of Zernike's ingenious transformation. The imperceptible phase variation is converted into a readily observable amplitude variation, making the transparent specimen visible. The mathematical description of this process involves the superposition principle of wave optics, where the total wave amplitude at any point is the sum of the individual wave amplitudes. In the case of phase contrast microscopy, we have two main components, the undiffracted wave and the diffracted wave. The phase plate modifies the phase of the diffracted wave, altering its superposition with the undiffracted wave. The resulting intensity pattern, which can be described using trigonometric functions, cosine and sine specifically, representing the wave's amplitude as a function of position, directly reflects the phase variations introduced by the specimen. A simplified mathematical model, while omitting many complexities of real optical systems, can illustrate the fundamental principle. Let's represent the undiffracted wave by a complex exponential function, e omega, where omega represents the angular frequency. The diffracted wave, after passing through the specimen, can be represented by aei omega plus phi, whereas the amplitude in phi is the phase shift introduced by the specimen. The phase plate introduces an additional phase shift, say, psi, to the diffracted wave. The resulting superposition of the modified diffracted wave and the undiffracted wave can then be analyzed to determine the resulting intensity pattern. The intensity, proportional to the square of the amplitude, reveals the contrast generated by the phase shifts. Calculations involving trigonometric identities, specifically those involving cosine and sine functions, are essential in determining the final intensity distribution. The specific design of the phase plate is critical to the success of the technique. The plate is typically a thin transparent disk with a central region that alters the amplitude and phase of the light, while the surrounding area remains largely unchanged. The precise dimensions and optical properties of the central region are carefully chosen to optimize the contrast and image quality. The challenge lay in engineering a phase plate that would introduce the precise phase shift required while simultaneously minimizing unwanted effects such as diffraction and scattering. Zernike's masterful design addressed these challenges, laying the groundwork for the widespread adoption of phase contrast microscopy. Zernike's work profoundly influenced subsequent developments in optical microscopy and wave optics. His mathematical treatment of phase contrast provided a rigorous framework for understanding and improving optical imaging techniques. The principles he elucidated remain fundamental to modern wave optics, finding applications in diverse fields ranging from optical engineering to materials science. The phase contrast method's enduring relevance in biological research and clinical diagnostics stands as a testament to the power of his elegant solution to a seemingly intractable problem. His work continues to inspire innovation in optical imaging technologies, pushing the boundaries of what we can see at the microscopic scale. The enduring legacy of Zernike's phase contrast microscopy is a testament to the power of combining deep physical insight with precise mathematical modeling. Building upon his theoretical breakthroughs, Frits Zernike embarked on a series of meticulous experiments to demonstrate the efficacy of his phase contrast method. These experiments were pivotal, transforming a theoretical concept into a practical tool with profound implications for biological research. Zernike's experimental approach was characterized by a remarkable blend of ingenuity and precision. He recognized the need for carefully designed optical components to manipulate the phase of light waves effectively. Central to his method was a phase plate, a crucial element introduced into the optical path of the microscope. This plate, subtly altering the phase of the diffracted light relative to the undiffracted light, allowed for the visualization of transparent specimens. The design of these phase plates was far from trivial. Zernike meticulously calculated the precise thickness and refractive index variations needed to achieve optimal contrast enhancement. He experimented with various materials and fabrication techniques, striving for consistency and accuracy in the phase shifts introduced by the plates. This attention to detail was critical, as even minor imperfections could compromise the effectiveness of the method. His early work involved painstaking adjustments to the optical configuration of the microscope, optimizing the alignment of the condenser and objective lenses to ensure that the diffracted and undiffracted light waves interfered constructively or destructively, depending on the desired contrast. One of Zernike's early experimental successes involved the imaging of microscopic items, single-celled algae with intricate, siliceous shells. Before the advent of phase contrast microscopy, these structures were challenging to visualize clearly. The delicate details of the item's shell were often obscured by the lack of inherent contrast. Zernike's method, however, revealed these features with unprecedented clarity. His published images showcased the intricate patterns and textures of the diatom shells, demonstrating the remarkable resolving power of his technique. These images were not merely aesthetic achievements. They served as compelling evidence of the technique's ability to reveal fine structural details previously invisible with conventional microscopy. The clarity achieved was a direct result of the precisely engineered phase plate and careful alignment of the optical components. His experiments extended beyond items, encompassing a wide range of biological specimens. He meticulously documented his findings, providing quantitative data on contrast enhancement and resolution improvements achieved with his method. These experiments confirmed the versatility and effectiveness of his phase contrast technique across various biological samples. The ability to visualize living cells without the need for staining was a particularly significant advancement. Conventional staining techniques often killed or altered the cells, limiting the observation of dynamic cellular processes. Zernike's method allowed for the observation of living cells in their natural state, providing unprecedented insights into cellular dynamics, motility, and morphology. The immediate impact of Zernike's work was felt across various biological disciplines. Cell biologists gained a powerful new tool for studying the intricate structures and functions of cells. Microbiologists could now visualize bacteria and other microorganisms with greater detail, leading to a deeper understanding of microbial structure and behavior. The ability to observe living cells in real time opened up new avenues of research, enabling the study of cell division, intracellular movements, and other dynamic processes. The medical field also benefited, with phase contrast microscopy rapidly finding applications in clinical diagnostics. The enhanced visualization capabilities facilitated the identification of pathogens and the diagnosis of various diseases. The instrumentation Zernike employed was, for its time, relatively simple. The core components were standard optical elements, condensers, objectives, and phase plates with a precise design, and assembly of these components were crucial. His experimental setup involved careful adjustments to the position and alignment of the lenses and phase plates to optimize the interference effects. This meticulous approach was essential for achieving the high contrast and resolution he demonstrated. The relatively straightforward nature of the apparatus, however, ensured the method's rapid adoption by other researchers. The accessibility of the technology played a crucial role in its widespread dissemination and impact. The influence of Zernike's experimental methodology extends far beyond his initial work. His emphasis on precise control over light wave phase continues to shape the development of modern microscopy techniques. His approach, emphasizing careful manipulation of light wave interference, directly influenced the development of differential interference, contrast microscopy, and other advanced imaging techniques. The rigorous experimental methodology he established provided a model for future generations of microscopists, emphasizing the importance of precise control over experimental parameters and meticulous data acquisition. The legacy of Zernike's experimental work is not confined to his published results. It extends to the broader scientific culture, promoting a rigorous approach to experimental design and implementation. The impact of his phase contrast microscopy resonates powerfully in contemporary biological and medical research, a testament to the enduring power of his innovative approach. His precise experimental methods, coupled with his theoretical insights, transformed microscopy from a tool with limited capabilities into a powerful instrument capable of revealing the intricate details of the microscopic world. This legacy remains central to the advancement of optical microscopy, underscoring the enduring value of his contributions. The influence of his experimental work continues to shape the development of advanced imaging techniques, ensuring that his legacy will persist for many years to come. Building upon the foundation of Zernike's meticulous experimental approach and theoretical understanding, his invention of phase contrast microscopy ushered in a transformative era for biological sciences. The ability to visualize living cells without the need for staining, a previously insurmountable obstacle, opened unprecedented avenues of biological inquiry. This innovation fundamentally altered the landscape of cell biology, microbiology, and related fields, revealing intricate cellular processes previously hidden from view. Before Zernike's breakthrough, the study of living cells relied heavily on staining techniques. These methods, while providing contrast, often killed or distorted the cells, limiting observation to static snapshots rather than dynamic processes. Zernike's phase contrast microscopy elegantly circumvented this limitation by exploiting subtle differences in refractive index between various cellular components and their surroundings. The technique rendered transparent structures visible, allowing researchers to observe living cells in their natural state. This capability proved revolutionary. The impact on cell biology was immediate and profound. Researchers could now witness cellular division in real time, observe the intricate movements of organelles within cells, and study the interactions between cells and their environment. A wealth of publications emerged, showcasing the newfound possibilities. For instance, studies on the dynamics of mitosis and meiosis provided significantly refined understanding of these fundamental processes. The detailed visualization of cytoplasmic streaming in plant cells offered compelling insights into intracellular transport mechanisms. Early work on bacterial motility using phase contrast microscopy provided crucial information on flagellar function and bacterial chemotaxis, illuminating the mechanisms behind bacterial movement and response to chemical stimuli. The implications extended far beyond basic cell biology. Microbiology, a field heavily reliant on microscopy, experienced a similar renaissance. The ability to observe living microorganisms without staining allowed for detailed studies of bacterial growth, morphology, and interactions with host cells. This led to breakthroughs in understanding pathogenesis and the development of novel antimicrobial strategies. Phase contrast microscopy also played a pivotal role in virology, enabling the visualization of viruses and their interactions with host cells, contributing significantly to the understanding of viral replication and disease mechanisms. The visualization of living cells in their natural state also facilitated advancements in immunology. Researchers could now directly observe the interactions between immune cells and pathogens, leading to a more nuanced understanding of immune responses. Studies on phagocytosis, the process by which immune cells engulf and destroy pathogens, benefited greatly from the ability to observe these events in real time. Phase contrast microscopy revealed the intricate choreography of immune cell interactions, providing a visual basis for mechanistic understanding. Consider the work of Albert Claude, a pioneer in cell biology, who extensively utilized phase contrast microscopy in his studies of cellular structures and functions. Claude's groundbreaking work on the endoplasmic reticulum and other cellular organelles, much of it facilitated by phase contrast microscopy, significantly advanced our understanding of cellular organization and function. His research, published in a series of influential papers, directly benefited from the clarity and detail afforded by Zernike's invention, contributing to Claude's eventual Nobel Prize in physiology or medicine. This is but one example of the myriad of researchers whose work was profoundly impacted by the invention of phase contrast microscopy. The enduring significance of phase contrast microscopy is evident in its continued use in modern biological and medical research. While newer, more sophisticated microscopy techniques have emerged, phase contrast microscopy's simplicity, affordability, and effectiveness ensure its continued relevance. It remains an essential tool in many laboratories worldwide, a testament to its enduring value and the ingenuity of its inventor. Its influence extends beyond its direct applications. It served as a springboard for the development of other advanced microscopy techniques, such as differential interference, contrast microscopy, and confocal microscopy, techniques that build upon the fundamental principles pioneered by Zernike. The legacy of phase contrast microscopy is not merely a historical footnote. It forms a crucial chapter in the ongoing narrative of biological discovery. It stands as a powerful illustration of how a single invention can fundamentally reshape a scientific field, driving decades of progress and continuing to inspire innovation in the present day. The ability to observe life's fundamental building blocks with unprecedented clarity, a capability Zernike gifted to the world, remains a cornerstone of biological investigation. The detailed observations made possible by his invention continue to shape our understanding of the living world, a testament to the enduring power of his innovation. Frits Zernike's invention of phase contrast microscopy represents a pivotal moment in the history of optical science, a testament to his profound understanding of wave phenomena and their application to biological imaging. His work transcended the limitations of conventional microscopy, opening a new vista on the microscopic world. The ability to visualize transparent biological specimens, previously obscured by their lack of inherent contrast, revolutionized cell biology, microbiology, and pathology. Zernike's achievement was not simply a technical improvement. It fundamentally altered the way scientists interacted with the microscopic realm. The impact resonated deeply, extending far beyond the immediate applications of his invention. Consider the sheer elegance of Zernike's solution. He ingeniously manipulated the phase differences of light waves passing through a specimen, converting these cell variations into readily observable intensity changes. This seemingly small modification, the introduction of a phase plate in the microscope's subjective lens, yielded a dramatic improvement in image contrast. His work directly addressed the long-standing challenge of visualizing transparent structures, such as living cells, which lacked sufficient contrast for observation using conventional bright-field microscopy. Before Zernike's invention, scientists were often limited to staining techniques that could alter or even kill the specimens under observation. His technique allowed for the observation of living cells in their natural state, providing unparalleled insights into cellular processes. The precise methodology Zernike employed involved a deep understanding of diffraction and interference phenomena. He carefully designed the phase plate to introduce a specific phase shift to the diffracted light relative to the undiffracted light. This manipulation enhanced the contrast between the specimen and its surroundings, making previously invisible details readily apparent. The mathematical framework underlying his invention was sophisticated, drawing upon the principles of wave optics and Fourier analysis. His contributions to the field of wave optics are significant in their own right, extending far beyond the specific application to microscopy. These principles remain central to the field of modern optics, underpinning numerous advancements in imaging and optical technologies. Zernike's influence on subsequent generations of scientists is undeniable. His work directly inspired the development of differential interference contrast DIC microscopy, a technique that enhances contrast by exploiting the differences in refractive index within a specimen. DIC microscopy, which relies on the principles of polarization and interference, builds upon Zernike's foundational work, extending its capabilities to visualize even finer details in transparent samples. Similarly, the development of confocal microscopy, a technique that uses a point-scanning laser to create high-resolution images, owes a debt to Zernike's pioneering work. Confocal microscopy, while employing different principles than phase contrast microscopy, shares a common goal of enhancing the visualization of microscopic structures, a goal Zernike first achieved in a remarkably innovative way. The modern relevance of Zernike's phase contrast microscopy remains significant. The technique is still widely used in biological research, medical diagnostics, and educational settings. Its simplicity and effectiveness continue to make it a valuable tool for studying a wide range of biological specimens, from individual cells to complex tissues. Researchers continue to use phase contrast microscopy to investigate cellular processes, study microbial growth, and diagnose various diseases. The technique's enduring utility is a testament to its fundamental importance and the elegance of its design. It stands as a shining example of how a seemingly simple scientific innovation can have a profound and lasting impact on the world. Beyond its direct applications, Zernike's work has had a tremendous influence on the development of optical science and technology. His contributions to wave optics have profoundly impacted various fields, from astronomy to telecommunications. The principles he elucidated continue to inform the design and development of advanced optical systems, shaping our understanding of light and its interactions with matter. His legacy extends far beyond a single invention. It represents a paradigm shift in our ability to visualize and understand the microscopic world. His work continues to inspire researchers and engineers, driving innovation and progress in numerous scientific disciplines. In conclusion, Frits Zernike's legacy extends far beyond the immediate impact of his phase contrast microscopy. His work represents a profound contribution to optical science, a testament to his ingenuity and deep understanding of fundamental physical principles. The enduring relevance of his invention, its influence on subsequent developments in microscopy, and its broader impact on optical science solidify his place as a towering figure in the history of scientific innovation. Zernike's achievement serves as a powerful reminder of the transformative potential of scientific discovery and the enduring power of elegant solutions to complex problems. His invention continues to enable fundamental advancements in biology and medicine, a lasting tribute to his vision and scientific excellence.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.